Hello, and welcome to Meet the Authors. Today we're hosting our newest session of the series, an opportunity to hear from and engage with authors of publications that were enabled by the Open Science Data Repository. For those of you who are new to this series, our format will involve a brief overview of the publication by each author, followed by a few questions from me. Then we'll open the floor for the authors to converse with each other or to respond to questions from you, the audience. Today's Meet the Authors features Dr. Jang-Hoon Kim of Weill Cornell Medicine, Dr. Fei Wu of Buck Institute, Dr. Emmanuel Gonzalez of McGill University, and Dr. Debbie Vero of the University of Miami. Dr. Kim, could you please start us off by letting us know a little bit about your publication, Single Cell Multiome and Immune Profiles of the Inspiration4 Crew Reveal Conserved Cell Type and Sex-Specific Responses to Spaceflight?
Sure. Hi, everyone. Thanks for this opportunity to share our work and also the introduction. Our study followed the four inspiration for civilian astronauts with a single-cell multi-omics lens before and after their 3-day orbital mission. We combined single-cell multi-omic transcriptomics and epigenomics analysis with immunoreceptor profiling and circulating markers to map how each immune cell type responds and recovers. We see a rapid coordinated immune change right after landing that largely resolves, but with cell type differences and sex-specific patterns in magnitude and recovery. Several signals replicate across prior astronaut and model datasets, pointing to conserved spaceflight biology and giving us concrete targets for monitoring and countermeasures for future missions such as Moon and Mars crews.
Thank you, Dr. Kim. Dr. Wu, could you please give us a brief highlight of your publication? Single cell analysis identifies conserved features of immune dysfunction in simulated microgravity and spaceflight.
Yes. Hello, everyone. Glad to have this opportunity to share our research. In our published paper, we across spaceflight and its analogs, we had profiled immune cells at a single cell resolution and found a conserved profile changes in stress or inflammation pathways, which were mainly due to the changes in cytoskeleton and antigen presentation cells. Those immune cells shows a shift that resembles accelerated immune aging. Then we further tested a compound called quercetin and observed a partial recovery of those dysregulated pathways in immune cells in vitro. So our paper also suggests feasible countermeasures that could stabilize the immune homeostasis during long-duration spaceflight missions.
Great. It's exciting. Thank you, Dr. Wu. Dr. Gonzalez, could you give us an overview of spaceflight alters host-gut microbiota interactions?
And about gut microbiome, so quite different from what we heard, and see how the spaceflight affects the gut microbiome and the host using mice on the International Space Station for up to almost 2 months. And we use high resolution methodology, big shifts in the gut bacteria species, and several important that you can call good guys are turned down while potential harmful bacteria go up. And, what was in a way surprising, lined up very closely with the gene expression changes in the colon and liver. And we're seeing bile acid regulation, energy metabolism, immune pathways affected. And even so, like disruption in the circadian rhythm and immune suppression signals. So quite a big story to tell there. And so in the end, this really shows how the gut microbiome and host metabolism are tightly linked in space. And just a quick note that there was 15 people working on that, so all coming from the GeneLab International Working Group microbes. And I've been there 4 years, I think, yeah, 4 years. I joined together with my pal Nick Burton, and it's a team effort. And we had three students, high school students from the GeneLab high school, high school program that did a great job analyzing the RNA data set.
Thank you for sharing that piece about your collaboration. I thought it was inspiring that these high school students were able to contribute, and you just met them at ASGSR, and it was just a great fortuitous thing for all of you involved. So we on all our fronts are really appreciative that you were able to take these on and recognize that they were able to contribute to your work.
Absolutely, they did a great job.
So thank you. And Dr. Del Rivero, could you please let us know about the effects of spaceflight on inflammasome activity in the brain of mice?
Yes, thank you. Thank you for the opportunity. And in this study with my collaborator, Dr. Upal Roy from University of Texas, Rio Grande, we studied the inflammatory response in the brain of female mice that were 37 days in the International Space Station as part of the Rodent Research 1 mission, RR1. And basically, we wanted to see, how this inflammatory response is regulated, or it's the protein expression levels in the brain of these mice compared to the ground controls. And we were hypothesizing initially that the inflammatory response of mediated by the inflammasome and other inflammatory cytokines was going to be elevated as a result of spaceflight in the brain of these mice. But to our surprise, they were actually downregulated, which was actually quite an interesting observation that we were not expecting at first. But there is some evidence that indeed there is a stalling of the inflammatory response as a result of spaceflight. So I'm happy to talk about this further.
Yeah, looking forward to that piece. So now that we know a little bit about each of these publications and the common threads, the surprises, and the corroborations, we see these common threads regarding immune function under spaceflight conditions. And so I'd like to begin with a few questions, starting with Dr. Kim. So it was interesting to hear about the immune changes across the different studies that echo the results from the previous NASA missions and even the mice flown in space. What does that say about how universal these effects are based on your research?
Yeah, great question. The short answer is these effects looks universal at both the gene and pathway levels. In our inspiration for single cell data and in prior NASA astronaut and other space flight studies with collaboration with Dr. Fei Wu and Dr. David Furman and Dr. Daniel Weiner's team, we repeatedly see the same direction. So mitochondrial oxphos and ribosomal translation go down, stress inflammatory program rise and then resolve, and antigen presentation tones down. The modules line up across cohort and species. That convergence gives us confidence we are measuring a core spaceflight response we can monitor and target, and likely conserved because shared stressors such as radiation or microgravity or circadian change or mitochondrial change push the same modules. We need more additional study to getting into the details.
Additional studies, that sounds that sounds promising. I have another question, but I want to come back to that after I hear back from a couple other authors. So I'd like to ask Dr. Wu. It seemed like one of the exciting parts about your study was that the natural compound you mentioned, quercetin, it seemed to help with the abnormal immune pathways. Could you actually speak a little bit more about that?
Oh, yeah, sure. Actually, in the aging research field, quercetin is well known as a senolytic, which targeted to kill senescent cells and reduce the SASP, which stands for senescent-associated secretory phenotype. Here, in this study, we found that under microgravity or spaceflight stressors, the quercetin targeted a broader range of inflammatory states. We saw it dampen the pro-inflammatory gene expression and partially restore the cytoskeleton and cell adhesion and antigen processing signatures. So I think the takeaway message is not quercetin as the answer, but it's a safe pleiotropic compound can shift the multi-omic signature changes in space, make them towards a here's a baseline. And this method also open a gate for screen more compounds on a larger scale.
Great,
This is my thought. Yeah.
I'm sorry for interrupting, but I think it's important to point out it's not like a cure or a catch-all, but it is one opportunity. My third question is for Dr. Gonzalez. Thanks for reconnecting. So I hope you're okay if I ask you a question. The study showed that the changes in the gut bacteria are tied to the shift in the immune-related genes in the body. So can you tell the public, why is that gut immune connection so important, and what could it mean for keeping astronauts protected from potential illness during long missions?
So what we saw in spaceflight is the barrier the gut barrier is challenged from two sides at the same time. And we saw genes tied to the gut barrier integrity were turned down during spaceflight, and things like extracellular matrix and IgA pathways were downregulated, and those are supposed to protect the gut lining. So it's pretty important. And it's a bit like scratching your skin. Normally, the skin is a solid shield, but once it's weakened, then it can create an entry point for infection. And on the gut side, many of the good guy bacteria, so bacteria that are usually in a healthy gut microbiome, were also turned down. And those bacteria, some of them, and we had two groups. So as I said earlier, the bile acid and butyrate producers were, for those who don't know, these are bacteria whose job is to keep the gut lining intact. And so they went down, and also at the same time, potentially harmful bacteria like C. difficile went up, and significantly. So it's not the best scenario, and it's a kind of double hit where you have weakened defense on one side and more pressure from opportunistic microbes on the other side. And so what we saw was a complex interplay. You had a compromised barrier, fewer protective bacteria, and more stress on the system. And on the other side, the host side, immune genes being dialed down. So this gut liver axis seems to be incredibly important. And for astronauts, this means they could be more vulnerable to infections or have slower recovery, and especially on long missions where medical assistance is quite far away.
Right, thanks. Do you think we need to start monitoring astronauts' gut health like the way we do for heart rate or bone density? Do you think that needs to be in place?
Yeah, I think monitoring astronauts' gut health would be a great first step. But it's not without challenges, and we are specialists, and we do this all the time in different setups, and every time we look at it, it's such a complex environment, ecosystem. So gut microbiota is not really tracked right now, but it definitely will tell a lot. And if we don't keep an eye on that, we're missing any early warning that the gut microbiome will tell us. But again, it's not without challenges.
Right. Wonderful. I'd like to come back to Dr. Del Rivero. So we've looked at some of the other studies. They've looked at mostly spaceflight effects on the immune responses throughout the body, but yours really takes a closer look at how the brain's own immune system reacts in terms of inflammation and neurological health. Could you speak a little more on how spaceflight seems to suppress the inflammatory response in the brain, that pause that you mentioned in your paper? And I'm curious, does it ultimately protect the brain, or does it leave it more vulnerable to infections or other stressors?
Yeah, so that's actually our current working hypothesis, where we were hypothesizing that consistent with previous studies that have shown that the inflammation is part of the innate immune response, and there are studies showing, for instance, that proteins of the innate immune response, like the STING signaling pathway, when they undergo stress, there is a stalling in replication forks. So it is possible that as a protective mechanism, there can be this stalling of the transcription translation of inflammatory proteins and other downstream cytokines uh, uh, with the intent of actually, yes, inducing this protection. The the interesting thing, as you mentioned, yes, this was in the brain, and there is evidence that the inflammatory response is not regulated as a result of a space flight in certain circumstances. But we've done follow-up studies in the Kennedy Space Center in the microgravity core facility over there, and although we haven't published the data yet, what we're starting to see is that indeed, there is a difference between what you see when you study cells, immune cells from the central nervous system compared to immune cells from the peripheral system. So definitely they behave differently. And yes, we believe that at this point that it is a protective response. However, whether it is long lasting after they come back to Earth, that's a different story that will be nice to look at it in the long term, because it could be that it's being preserved or conserved or slowed down this production of inflammatory proteins. But once they come back to Earth, and then as they continue to acclimatize to the normal environment down here, then that inflammatory response may start to exacerbate tremendously. That's something that hasn't been determined yet. But this idea of lowering of inflammatory proteins in the brain, it's the opposite of a concept that we call inflammaging, which is age-related inflammation, or inflammation as a result of aging that occurs naturally. And what happens is that as we age, obviously our brain and our body starts to get inflamed. That didn't happen obviously in the mice that went to space, and that's also consistent with previous data that have shown, for instance, in the twin study, where the twin that was in the ISS actually aged at a lower rate than the one that remained on Earth. So there might be a story there in regards to decreased inflammaging uh, and as a result of space flight. So we'll see.
We'll see indeed. I mean, it's exciting. It's giving us a lot to look forward to, right, as we keep an eye out for that latest research that's coming out. I wanted to circle back to the question that I had for Dr. Kim earlier. So I'm curious, based on your research, how might that help shape planning for future space travelers or future astronauts? Because now I feel like there's some connections going on here.
Yeah. So as I introduced, the signals are conserved across the tissue and cell types and multiple missions and species. We can move forward, measure everything to our standard playbook. So we can start from the baseline and design an in-flight minimal panel and checking the landing spike. So crucially, also we can translate from model systems to human. Mouse and organoid studies are cheaper, accessible, and modifiable and scalable, so we can pre-test OXPHOS, mitochondrial, and antigen presentation countermeasure, or microbiome circadian things, then port the winners to the astronaut. So also the cross-study convergence let us plan care that's both standardized and personalized.
Great, thank you. And to pair these two ideas together now, and I think both of you have now touched upon them, I'd like to ask both Dr. Kim and Dr. De Rivero, do you expect more studies on duration-based effects or for those longer-term situations like you mentioned? Do you think there's a threshold where you'll see these effects that describe what really happens? And feel free to go first, Dr. Rivero.
Sure. So I think, since the goal is to go to Mars, I think those studies are granted, right? So definitely we need to understand how those changes happen as a result of much longer explorations. So we need to start considering monitoring astronauts better for these long intended flights, and also obviously rodents along the way, and try to correlate one with the other to better understand what's going on. But especially because when you think about what happens in the immune response within the brain, for instance, the blood-brain barrier, which is the barrier that divides the brain from the rest of the body to maintain it in a sort of like what we used to call immunoprivileged status, it gets affected as a result of spaceflight. But even in disease, for instance, brain trauma, when the blood-brain barrier gets affected, it's not that gets affected continuously, but it's an asynchronous process. So you need to actually have different time points to start looking into these different things, to look at the, let's say, inflammatory response throughout the longer exploratory procedures. Fortunately, nowadays we have instruments where we can actually detect in blood proteins that are found normally in the brain. So we have sensitivity enough to find in blood things that before we couldn't, because they were not. We didn't have the technology of the sensitivity to measure brain-derived things in the blood. We used to just do it before in the cerebrospinal fluid, but now we can do it in the blood. And I think we can incorporate these studies in these longer duration flight studies.
And go ahead, Dr. Kim, to address that as well.
Yeah, sure. I can continue. So, absolutely, duration and dose matter, and we already see distinct regimes. In our work, we found that some molecular immune signatures line up between a 3-day flight from Inspiration 4 mission and 1-year class mission, the NASA Twin Study, despite the short flight not producing the biggest physiology shift, suggesting conserved biology that scales with time aloft. And also, it is evident there is a timeline of physiological changes upon duration. So, from hours and days, there are some rapid fluid shift headward, and the plasma volume drop in 24 hour, and space motion sickness is also kind of acute changes. And during the 1 to 4 weeks, there'd be kind of early muscle atrophy and rising bone resorption markers. And kind of 3 to 6 months, there'd be kind of cumulative bone loss and persistent immune dysregulation and latent virus reactivation, or space-associated neuroocular syndrome. And kind of 1-year mission, there'd be a broader changes related to mitochondrial adaptation and some slower resolving changes. So looking ahead, duration drives designs. Artemis 3 is a kind of 30-day end-to-end with a week on the lunar surface. Mars plans assume 6 to 9 months, or be longer than 9 months. It would be kind of 500 days. So expect non-linear threshold once crews cross time on orbit and sleep disruption limits. And with International Space Station operating to 2030 and commercial low Earth orbit stations such as Axiom or Starlab or Orbital Leap or VAST coming online, we can finally run duration stratified protocols, short, medium, and long, without being bottlenecked by one platform.
It's really cool to hear how you guys are able to communicate with each other about this, even though you have, somewhat different topics in your original publication. So there's like a recurring theme. I have a question to pair Dr. Wu and Dr. Gonzalez now. Could you actually address the value of these kind of cross-study discussions, especially, as we say, with Moon and Mars goals for space exploration? Dr. Gonzalez, feel free to go first.
Yeah, I mean, those cross-studies discussions are absolutely essentials. And, there's not a single experiment in space that has enough replication to answer an important question. And when we compare across different missions and data sets, we can see consistent patterns. And that's where you gain confidence in your own results, I would say. So, for example, in our mouse study, we clearly saw disruption in the gut microbiome and immune-related pathways. But what was striking is that when we were working on an article 2 years prior to this one, and about the Mars 500 experiment, we saw similar kinds of disruption, especially in glucose regulation and gut inflammation markers. And Mars 500 was simulating long duration confinement for humans. It was on Earth. And so even though it wasn't actual spaceflight, we saw microbiome and immune shifts that were echoing pretty well what we saw here with animals after spaceflight. We saw disruptions of the similar bacteria in patient, fibromyalgia patients, so chronic pain patient also. So when we saw those bacteria, we were like, oh my God, but we know those ones. And so, when you're doing this kind of work, you always, you have to be a little bit paranoid because you don't know what if you're going to, or if you're overlooking your experiment, you might say some stupid things, and you don't want those to be published. But when you see, or when you discuss with colleagues, especially here with single cell or people that are doing immunity, for their work, when they see the same thing, it's kind of reassuring. So it's like bringing these threads together. It's just incredibly valuable.
Yeah, I agree with the point Dr. Gonzalez just bring out. I think we needed to find a better way to harmonize and combine the different analysis in database across so many studies in the field. All this method, we find a better way to do the meta-analysis of these studies. There are huge value would coming out. For example, for us study like analyze cross single cell and microbiome immune studies in this small area. But if we can combine these two type of studies, we can pinpoint the truly conserved biological underneath those changes and can reduce the study-specific artifacts. So practically, I think it can accelerate the countermeasure compounds development. For example, if a pathway is conserved in both immune and the gut immune axis, it can be moved up the priority list for the in-flight monitoring or intervention later on.
I think you raised a really important point about how it accelerates these kind of studies. We have these conversations and, we've got attendees who might be working on similar research who might have just felt validated by what both of you have said. For each of you, what do you feel like is the call for action from the space flight and even the Earth community, or really the public in general, for the importance for addressing how space affects the immune system? And if you really want to tie it to the terrestrial applications, feel free. But my question is, what do you think is the call for action from the spaceflight community and research? Let's start with maybe Dr. De Rivero, and we'll work our way around to Dr. Gonzalez, Dr. Kim, and Dr. Wu.
Sounds good. So I think we should continue working as a team. This community does an excellent work working and analyzing data and designing experiments as a team, given the opportunities to actually run experiments in space. But we should be mindful that just by understanding how the immune response is modulated in space, we may be able to identify therapies to treat patients down here for, let's say, neurodegenerative diseases. So, for instance, if indeed situations associated with spaceflight are able to slow down the inflammatory or decrease the inflammatory response in the brain, then we could apply similar approaches on Earth to actually lower the inflammatory response that we know contributes either to the onset or the progression of neurodegenerative diseases or things associated with brain trauma, for instance. So there's a lot of benefit that can be gained as a result of this continuing understanding of how the immune response is modulated due to spaceflight.
Okay, I would like to say 2 points. First, I think we need in future we need to standardize the immune and microbiome sampling across missions, so we can combine and aggregate those data in a large scale, and also benefit even broader community to do the analysis. Um, and second, I want also hope we can develop a fast or low burden immune surveillance method can be used in flight. For example, we can learn the technology in the field like a point of care, and which is largely applied during the pandemic. And like for example, we can build another small easy use panel to measure the immune health, and this can be used uh, on the spaceship by the astronaut, for example, and to test the pre-approved countermeasures. So we have tested before and working as a monitoring method for rapid testing and feedback.
Yeah, I think I can continue. I'm echoing Dr. De Rivero, and I'd ask the community and the public to help us open the doors and work together. So it'd be kind of outside of immune focused call. It's more broader call. So as government programs and private flights accelerate, we should harmonize sampling and share by specimen and data and co-analyze using transparent reproducible pipeline. This is just the same one the OSDL community is pursuing. And right now, too much remain compartmentalized across agencies, countries, and companies, which slows discovery. So for 3, 4 years, there are kind of similar programs launched using some a human astronaut or the different government or different prime mission or agencies, but uh, it's kind of similar pipeline, but data is not shared very easily. So and the kind of this kind of field is limited with number of samples. So yeah, I think it'd be great to have a good way how we can collaborate together. And if we commit to shared core and common assays and federated data access, we can compare short, mid, and long duration mission directly, test countermeasure faster, and translate model system findings to human more reliably. So the immune system is mission critical, so I think we can take on the bigger challenge ahead based on this idea.
All right. Yeah, I was also going to talk about the community and the importance of it, but coming next and last, I'll change my perspective to towards microbiome. I think when you talk about immunity, you don't really think about gut microbiome, but we can't really separate immunity from the microbiome. And the microbes in the gut are constantly shaping how our immune system behaves. Again, think of it as an inner skin that we have. And so it's so important, this gut lining. And it is really tied with the host immune system. And we saw this connection so beautifully in our study. It was incredible. Now, the challenge is that microbiomes are incredibly individual. And when you think about, going to space or even on the ISS, but even more on the moon or Mars, you know from experience that two astronauts are from different cultural, dietary backgrounds, and they might start the mission with very different gut communities. And under space conditions like microgravity, stress, confinement, those differences in the gut communities could lead to very very different immune responses. And that means there's probably isn't going to be a one-size-fits-all solution. So, to me, the call would be to embrace that complexity rather than simplifying it. And space puts human in an environment we can't really replicate on Earth. But with modern tools, what we did here, multi-omics, and I would say the personalized microbiome analysis, this interpersonal differences in humans is quite a challenge for anybody working on microbiome. But when we look into this, so multi-omics plus personalized microbiome analysis, you can start to untangle how these complex systems interact with each other. And I see that, as the way forward. And treating astronauts' health as a complex, individualized system rather than assuming that the same countermeasure will work for everyone.
I think that's something that as you speak, I realize I've taken for granted. That we have a lot of sex-specific studies, we have a lot of these kind of things, but even just the diet from culture, I think that's something I've taken for granted. So I'm really thankful you brought that up. I'd like to open it up to the audience to ask a few questions. And so for the audience, please feel free to type into the chat for us to read your questions, or you may request to be called upon. And it looks like that Ryan Scott has a question that he would like to begin with, or that he could begin with. Ryan, do you want to unmute, or do you want me to read for you?
Yeah, it's pretty much Faye, I'd love to hear about your ML in your paper. And I know it had the flavanol and quercetin, but I'm just curious about any of the nuts and bolts of which model, why that was chosen, just because I sort of see the role of AI ML in the future playing more. And I was just thrilled that yours had that. And then the other question after that, I was about to write it into the chat, was like you sort of mentioned, Emmanuel, on precision methods. Well, Faye, maybe go first. My other question was on bioengineered crops, Emmanuel, for countermeasures. Like there was a recent AWG paper looking at plants and humans in space and gut health. And so especially the gut people here, I'd be curious if their thoughts on bioengineered crops and the pros and cons. Thank you, Jennifer.
Okay, thank you, Ryan, to ask those questions. It's a lot of aspect. First, I want to say generally speaking, to find the quercetin, we use our package called GCEA, developed by our colleague in the same group. Basically, the method is to first we curated through a large database, compound database, not only drug but also food and other related chemicals. And those data have the, for example, expression profile with the gene expression profile related to the in a cell, like after treated compound, the cell has been disturbed, and we can see the different profile, and we match those profile with our microgravity treated PBMC, and find those links. We use the facial exact test to see the similarity, and also hypergeometric test. These are very normal method to measure the previous data with the compound perturbation and our own immune microgravity perturbation. And we have received a long list. Not only the quercetin, but also a lot of other chemicals and different even food. And we rank them for just by the like a similarity adjusted p-value, and we first select some promising compound, also high ranked to test. Because quercetin is well known in the senolytics and dampen the SASP, because we know the SASP contain a lot of inflammatory factors. So maybe more related with our scenario in PBMC, and it works well. So it definitely was to test more candidate in our list. in future with a high throughput method. And also the generally and also in our single cell analysis pipeline, we including some method for the QC, which is also very important after those stressor disturbance of in the immune cell, which are very vulnerable. So the careful QC method is very important. So the machine learning there is like clustering and embedding, very general, often used to do a better QC in single cell.
Great, thank you. And Ryan also noted that Dr. Wu's paper is one of about 36 papers to leverage the AI/ML approach, algorithms towards the field of space bio and space health. So I wanted to point that out just to include that as well. We have another question from John.
Yes, thanks, Jennifer. Hey, first, thanks very much to the team. This was not only an informative briefing, but really opened up my aperture to some things I hadn't thought about. And the question I had was, listening to some of the discussion points about, I think it was Mr. Kim who talked about, calibration of data, aggregating and standardizing it. But it also feels like Dr. Wu had done, applying AI machine learning, but the challenge seems to be that the data is still siloed. And so my question is, as NASA starts to move away to embracing commercial space industry for them to deliver capabilities, what's the thought process? Should this something that NASA should take on as having a repository for human space in the space economy that's open to all, right? So it's based on open standards, but becomes, for lack of a better word, a playground for all that's data to live in, so that individual researchers or teams can come to the data and expose it to AI and analytics for the broader good. Thanks.
As the missions are getting commercialized, I guess it'd be a little bit tough to make them to share all the data as we did for the kind of government supported missions and uploaded in the GenLab or OSDL. So I don't have a very clear idea yet, but one of the way would be the commercial space flight companies could make a kind of big collaborating forum or kind of system, and maybe start to discuss about how we can make a synergy between the teams and also some government supported mission or some the other kind of private company supported mission. But so, and also for the private the commercial space flight mission is it's a kind of commercial and also private, so it's not kind of government astronauts. So it'd be for our prior experience it'd be a little bit limited to get approval, or sometimes the crews don't want, so there'll be another issue. So, and also it's very identifiable in some cases. So yeah, so I think we need to think about kind of a new structure or new system and how the commercial systems can collaborate to make a public goods. So it's very kind of vague for now, but yeah, maybe kind of think of a big forum on my side. And the other panelists, yeah, please fill in.
Yeah, and I think to address, so Open Science Data Repository does, of course, have mouse model organism, cell lines kind of data. And of course, that is all public. And as you've mentioned, as you noticed, some of these people mentioned the AWG communities, the analysis working group communities that have enabled them to have cross conversations about these kinds of, across these kinds of studies. So while you mentioned it could be siloed, but it's not entirely so in light of the things that Dr. Kim mentioned about some of the privatization and those kinds of abilities to share personal data, identifiable data. Ryan also mentions that a single database has been tried and it's tricky across those stakeholders and interests. And one solution might actually be federating across databases or across government, commercial, academic resources as well. So you raise a good point. And there are channels that are working with that. Would the other panelists like to chime in?
Actually, Jennifer, this is Ryan. John, yeah, your question is quite pertinent. That was sort of the purpose of why we even named OSDR the Open Science Data Repository, was to
Okay.
hopefully be that central type repository so that commercial, academic could all submit their data. NASA's still working on a lot of the legality, right? Because having all that inspiration for data there last year, there is some of the data that are there, but some of the raw reads, to be honest, the IACUC was unhappy with a lot of that these data were completely open, and so there's some internal discussion still about what's the best road forward. But that's why I think federating across databases might be right. You can just go to one portal, but then you can actually federate to where the data is housed. Like for instance, OSDR and GeneLab have a collaboration with the European Space Agency where their data will stay in Europe, but all the entries are federated with GeneLab and OSDR. So that's why that way the stakeholders will be happy. They're still holding on to their data, but the accessibility is now just being federated. So you're right on probably the most important topic for our entire field, John. So yeah, thank you for raising it.
No, well, thank you, Ryan. And I guess I'll have to file this under. It sounds simple, but it's not easy.
Yeah, there's a lot of missions, a lot of different funding mechanisms. Everyone wants to make sure that they are best situated, and it's global. So there's culture. There's handling IRB. Oh, I didn't mean IACUC. I meant the IRB was unhappy with the. Sorry,
Right.
I said IACUC as an animal. Sorry, but yeah. So there's just a lot of different moving pieces in this field, but we're my goodness, compared to where we were a decade ago, unbelievable progress has been made, but there's quite a bit more to be done. So thank you.
Well, thank you guys. Appreciate it.
Studying countermeasures, as each of us has mentioned, is a very hot topic right now. Could you just briefly address how we're filling in these gaps? Maybe Dr. Del Rivero and Dr. Gonzalez might want to address this one.
So, to be able to come up, it's like developing therapies, right? So to actually come up with countermeasures, you have to really understand what you're dealing with. So, for different situations or conditions, whether it's the inflammatory response in the brain, the periphery, or things like muscle atrophy, or things due to radiation or microgravity, all those different things create different problems. So it's since we're dealing with human lives, one has to be very mindful of what countermeasures to dare to try, and have a lot of actual knowledge behind trying the different countermeasures in terms of health, right? There are things obviously that can be done to make the life of astronauts up there even easier for them to do their mission, but it is tricky to come up with actually certain countermeasures for different things, since since the problem seems to be so multifactorial. It like when you try to treat multifactorial diseases, there's this concept of polypharmacy, where you try to come up with something that actually can treat a lot of different things at the same time. So that type of countermeasure might be actually needed eventually down the line. But obviously, first it's easy to focus on one problem and then goes and see how that particular countermeasure works for a problem. In the case of the brain, let's say, it would be nice to regulate with things that are available already, commercially available, to regulate different cytokines. So if a cytokine is elevated in the blood of astronauts, and we know those cytokines are not beneficial, let's say tumor necrosis factor, so then we can use anti-TNF that has been used safely for a couple of decades, and see if that makes life easier for these astronauts as a countermeasure. So there are alternatives that can be tested little by little, and see how that goes.
Yeah, I'll echo Pablo's answer about complexity, and it's countermeasures. It's simple to give a countermeasure here talking, but when you are actually modifying somebody's, let's say, gut microbiome, trying to protect the astronauts by stabilizing a microbiome, gut microbiome balance. It can be, it can be too simplistic, and there is not a turnkey solution to stabilize one's gut microbiome. And even the stability or balanced microbiome is a contentious notion in metagenomics. So it's, a countermeasure would be to test probiotics, prebiotics, even postbiotics, and dietary tweaks that encourage something like, in what we saw in our study, something like butyrate production. Butyrate is so important for a healthy gut lining. So, but these are simple words. Again, like I could say to somebody, to my neighbor, yeah, you should take prebiotics or have yogurts, eat diverse, but you never know. And when you have a complex environment, just like space, and where isolation takes a toll on the psychology of the astronauts, and the gut is so linked to the brain that you'll have multiple factor happening at the same time. It's something that is hard to to design effectively, and so we need more trials to do those interventions. Having for example, you send mice to the ISS like we did, and after a month you start to see those changing happening. It's very quick. It's incredibly quick that it happens, and then you can take those tools, transfer it on the ground to see whether there's a change, and do the same thing on the mice that are on in space. You can transplant this tool from ground mouse to see if you can reverse a little bit this destabilization. It's something like this, something where you start to work out new experiments. I think that is the best countermeasure right now, rather than saying something that's simple and plenty of void, like, yeah, we need good dietary habits and eat prebiotics. It's more complex than that.
Thank you for reminding us of the complexity. As we're approaching the top of the hour, I'd like to invite our panelists to give a short, less than 1 minute closing thought for each of us. And we'll start with Dr. Wu, then Dr. Del Rivero, Dr. Kim, and Dr. Gonzalez. So just a short closing thought for us to carry with us for possibly the rest of the day or through to our morning.
Okay, thank you. I think nowadays it's very promising to build a foundation model-based virtual cell, as it's able to predict how cell responds to a treatment by a method called in silico perturbation. However, they are mostly training on compounds and drugs, which missing the microgravity or space environment or context. So they can't be predict where what happened in space. Because we know microgravity shift immunology, so I believe that the future foundation models really need a large-scale microgravity perturbation data from organoid or PBMCs. So this is my thought.
Yeah, thanks for your time, and thanks for this chance again. And I'm in the Walkner Medicine and also Christopher Mason Lab, and our team is working on the Space Omics and Medical Atlas team, and also Cornell Aerospace Biobank, Cornell Aerospace Medical Biobank. So we have some astronaut collection and also the control data sets. So feel free to reach out if you have any interesting idea to test with the astro samples. We are happy to share the data and also the sample types. And also I recently joined the BioAstra, and BioAstra is a kind of non-profit organization and trying to make a kind of platform and environment and plan for some interesting spaceflight missions, and stay tuned about that one. And yeah, thanks again.
And Dr. Gonzalez.
Yeah, okay. So I think, what really comes out of our study is that the gut microbiome is not just about digestion. It's deeply tied to metabolism, immunity, And in space, that connection seems to be disrupted in a way that could matter a lot for astronaut health. And so the last message would be simple for me. If we want astronauts to stay healthy on long missions on Mars or the Moon, then the gut, liver, immune axis can't be ignored. And as the microbiome might turn out to be one of the keys to making deep space exploration possible.
Welcome back, Pablo.
Thank you. And I just wanted to add that we should continue doing this type of work because each mission is different, right? So if things are real, they should replicate from mission to mission, and we should learn something from each mission. But in the case of our study, it was from Rodent Research Mission 1. So it'll be nice to actually continue with the work to see how with more consistent missions and more similar missions and newer missions to see if the results hold along so that we can actually eventually say, yeah, this is probably what happens in humans in astronauts.
Thank you, panelists, for all your time today. And to our audience, if you were especially interested in any topics mentioned today, or if you had unanswered questions, please feel free to contribute to the conversations by joining our analysis working groups through the QR code that I'll display next, or the link provided here. And thank you, audience, for joining us, and please have a wonderful day.